do this one. This one's easy because you're just watching, right?
Hello?
Hello.
Hi.
Hi. We're just taking a bit of time to log in with the computer because we restarted the whole computer.
All right.
So we might take about 2 minutes until we properly join.
Okay, no problem. Just let me.
Ali?
Yep.
Quick question, Ali. You've already started it, haven't you?
He sent the link.
I haven't started going through the paper yet, but
No, but you've started the Hangout, haven't you?
Yeah.
Okay.
Yeah.
We don't need a new one. Yeah. Okay. Cool.
Yeah.
All you need.
We just need to log into the Slack.
Sorry. So we just go to Chrome, new window, and we'll just log into what's it called? CIE. These people are the reformat computers.
The thing is, the computer's
CIAS zcoach dot slack. Yeah, I'll do this one first. Enter your email address and your password. I'm going to sign in as me.
It was you before.
Yes.
Yeah.
And then Thomas. So I need to go and sign into all computers. Don't I? Brilliant. There you go.
Yeah. Okay. We're good. Okay.
So,
Hello.
hello? Hi.
Hello.
Can you hear me?
Yes.
Okay, perfect. Are we ready to go?
We have another student joining us today too, so we'll be 2 students.
Okay, sure.
Can you see?
Yeah, he can hear you.
Hello,
Hi.
I'm Sarah.
Sorry, could you repeat that?
I'm Sarah.
Sarah. Okay, hi. Nice to meet you.
Okay, where can I raise the voice?
Okay, are you guys ready to start?
Yeah, we're ready.
start, and then I'll get this one.
Okay, and you can all see the paper, right?
Yes, we can.
Okay, perfect. Let's get started.
No, I'm saying that I want to see. If it might be easier, then you have to start at Peter.
Okay, so let's start with question 1 then. Mark, can you answer the first part?
Okay. So the chamber that pumps blood into the pulmonary artery, that would be the right ventricle.
Yep. And Sarah, the second one?
A blood vessel that transports the oxygenated blood into a tube. It's the upper vena cava.
Yep, vena cava.
Thank you. The specialized tissue responsible for delaying the conduction. Yeah, so it's AVN.
Yeah, AVN. And which node starts the impulse?
The SAN.
Yep, good. Okay. D, Sarah?
Oh, the blood vessel that supplies cardiac oxygen. Wait, it starts with C.
Yeah.
It's I forgot.
It's okay. It's the coronary arteries. Okay. So in heart attacks, the coronary arteries are the arteries that get blocked up and cause ischemic damage to the muscle tissue. Mark, part E?
That would be the bicuspid valve or
Yeah.
the mitral.
Yeah, so you could say bicuspid / mitral / left atrioventricular. So you could use any of those terms and you'd get the mark for that.
Okay.
Will you get the mark if you say, oh, atrioventricular without?
You'd have to specify left.
Okay.
Because they're talking about oxygenated blood. That's the difference between the left and the right side of the heart.
Mm-hmm.
Okay. So have a read of that and tell me when you guys have read it.
Okay.
Okay.
Well, have you finished?
Yeah.
Okay.
Yes.
So before we go into this answer, it's a 3-part question. And we're going to describe the damage caused by this microorganism and then explain how it will affect the functioning of the epithelial tissue. So could somebody start by telling me what structure they think the microorganism is attaching to here?
What structure the microorganism is attached to?
Yeah. What are these?
The cilia.
Yeah, correct. So can you guess what kind of damage is being caused by the pertussis?
Okay. So, well, in the area labeled X, you can see that there's no presence of cilia. So, the bacterium has decomposed or destroyed the cilia of the epithelial tissue.
Yeah, good, that's right. So you get 1 mark for saying that the cilia are either absent or short. You could say that they're destroyed or damaged.
Mm-hmm.
Any other points that you can make there?
So that paired with the excess of mucus being produced makes it hard for the mucus to be transported up the airway, which is done by the cilia normally beating the mucus towards the larynx and then being swallowed down into the stomach acid.
Good.
Okay.
Yeah, so basically, I mean, you're going on to the explanation, which is good. There's 1 mark here for, as you said, talking about cilia being destroyed. You could also comment on the fact that it seems like there's some form of scar tissue replacing the cilia here in X.
Mm-hmm.
And obviously points 1 and 2 would affect the coordinated movement of the cilia. So if you talk about the idea of the cilia being unable to move coordinatedly, so unable to move with coordination. And then the other points are that lets the mucus accumulate, as you said. And the last point that you could make is that the bacteria is then trapped in the mucus and it's able to accumulate in the airway.
Yeah.
So pathogen increases in mucus. So those are like all the different points you could make on the question. But since it's just a 3-mark question, you don't need to hit all of these points. These are just the different things you could talk about.
Sure.
Are you guys happy with that? Okay.
Yeah.
Let's move on then. Cyril, can you answer this question?
It says produce mucus, maybe more structured, but also produces mucus. And because exchange system.
So there are mucus glands in the gas exchange system, and that's the answer here. So, other than goblet cells, you also have glands which are able to release mucus into ducts in the upper respiratory system and release it. So here the answer would be mucus gland. Mark, can you talk me through question C?
Yeah. Suggest how whooping cough is transmitted. Could I look at the picture again?
Yeah, cool. So,
Okay. Okay, it's transmitted similar to tuberculosis. Okay.
yeah,
So it's transmitted by infected droplets,
good.
with the bacterium being coughed or sneezed from an infected person.
Yeah.
And then those droplets entering the body of an uninfected person.
Yeah. So infected person coughs, and then infected person breathes in the droplet.
Yeah.
Sorry for my handwriting. But yeah, you guys happy with that?
Yeah.
All right. Next question, Stuart.
The process outline at the stage of the transcription. So first of all, the unfolding of the DNA, this is the first step.
Yeah.
And then the mRNA nucleotide.
Yeah, so the first thing we have is unwinding of the DNA. So which part of the DNA is unwinding?
Sorry, your volume is quite low right now.
Oh, I'll try and put it up.
Okay.
Let's see. Okay,
Oh, that's much better.
is that better?
Yeah.
Okay, sorry about that, guys. Okay, so the first step in production of
Sorry, again. Could you put it a bit quieter?
yeah, sure.
Okay.
Is that okay?
Yeah, that's okay.
Okay. Cool. So basically, what process is this answer looking for you to explain?
I didn't get what you say.
So what cellular process are you being asked to explain in this question?
It's the transcription.
Yeah, good, transcription. And so the first step of transcription involves reading the DNA, right? And it's a specific part of the DNA that's read. What part of the DNA is read in transcription?
Do you mean the gene or?
Yeah, exactly, the gene. So the part of DNA that's unwound is this gene, Mus5AC. So the first part is taking the DNA, which looks a bit like this, and unwinding it so it looks a bit like that. And this part is the gene Mus5AC. Okay. And here you'll have loads of bonds between the nucleotides. And to progress transcription, what do we need to do to those bonds?
Okay.
So first step is unwinding DNA. Second step, what do we need to do?
mRNA is gonna, what did I say? The mRNA is gonna be completed or complementary with the basis of the DNA. Is that right?
Yeah, that's right. But first of all, what we have to do is break these bonds. So these complementary bonds between the helices of the DNA need to break so that we can then fit in our enzyme to start synthesizing the mRNA. So you end up with something that's more like this. So these hydrogen bonds have now been broken. So that's your second step. So H bonds broken. And then the third step is that we're going to use one of these strands as a template. And for the sake of this diagram, I'm just going to say that this is the template strand. And when you're explaining transcription, you definitely want to make that point that one strand acts as template.
So it's a semi-conservative transcription, is that?
So it's the conservative nature of DNA replication is specific for DNA replication because you're talking about making new copies of DNA. Transcription is very different. Transcription, we're not making DNA, we're synthesizing mRNA.
Oh, yeah. I just get confused.
That's okay. That's fine. So after this stage where you've broken the bonds and you have your template strand, that's when the step that you were talking about happens. So you have RNA polymerase come in. And do you know what RNA polymerase does?
It unfolds the DNA. Or it starts from 3' to 5, something like that?
Yeah. So, but the main point here we say that RNA pol is this big circle, and it binds here, and it starts synthesizing mRNA, right? And the way it does that is it adds complementary RNA nucleotides to these bases.
Okay.
And that's what you were talking about. So complementary RNA. And that happens step by step. And the new sugar phosphate backbone is then sealed. And you have the product, which is the mRNA. And that's basically what the process is. For this question, they're only looking for you to make 4 points. But if you just work in sequence from the start, so start by saying that the DNA is unwound, and then talk about having a template strand and RNA pol coming in and synthesizing the mRNA by adding complementary nucleotides, then you'll get all the marks. Are you guys happy with that?
Yeah.
Okay, good. Let's do this last part of the question and then we can take a break. So, Mark, can you answer this?
Okay. Let's modify this. Okay, so glycosylation occurs in the Golgi apparatus.
Yeah, good.
And the mucin is packaged into ventricles to be exported with bulk transport by exocytosis, which requires energy out of the cell.
Okay, good. So, first point there for mentioning the Golgi apparatus. Second is correct. We're packaging them in vesicles to transport them to the cell surface.
Yeah.
You get another point for referring to bulk transport.
Okay.
And what do we use bulk transport for generally? Why bulk transport?
So why do we use bulk transport? Or
Yeah, why would the cell use bulk transport?
because the material is of a larger mass.
Exactly. So, mentioning that it's a large size which
Would
makes
have-
sense because you can imagine that the mucus produced is actually very large in volume. So that's why the cell uses bulk transport. And if it didn't use bulk transport, it would be very difficult to move it across the membrane.
Yeah.
Okay, good.
Answer the question. I don't know what's the answer to the question.
You don't know the answer.
I haven't come across this before.
You don't know the answer to E?
Yeah.
Okay.
Can you
Okay.
answer the questions?
Okay.
Okay, we'll talk through it. So, describe the signs and symptoms that enable diagnosis of COPD. So, Cyril, you're saying that you're not sure about this one?
I can give some suggestions, but I'm not sure about that.
Okay, go ahead.
Reproduction is one of the solutions. Oh, maybe shortness of breath.
Yep, shortness of breath is one.
And let's see. There should be some a squirrel sound when someone is sleeping or something like that.
Yeah, so a wheeze, is that what you're talking about? So wheeze sounds a bit like is that the sound that you're talking about?
Yeah.
Yeah, so that's one of the signs of COPD. Anything else?
And also obstructive pulmonary disease. Since it's pulmonary, it should be just shortness of breath, wheeze, chronic obstructive pulmonary disease. What else? Apart from doing with breathing, maybe he should have some heart problems.
Yeah, I mean, COPD is often accompanied with heart problems, but in this question, they're asking you to specifically describe the signs and symptoms that enable diagnosis of COPD. So they don't really want you to talk about the potential heart problems that a person with COPD might have. In terms of people who have COPD, do you know what one of the major causative factors of COPD is?
Smoking.
Yeah, good. And if you think about people who have been chronically smoking, can you think of any of their signs and symptoms?
Coughing.
Yeah, good. So chronic cough. Anything else?
Chronic cough. And what else? Let me see. Smoking. So they should have some teeth problems.
Yeah. I mean, that's correct, but that's more related to the effect of the smoke on the teeth as opposed to the effect of the smoke on the lungs. But other than that, you can have difficulty walking and exercising because you have impaired gas exchange, so you can't get oxygen to the muscles that require it, so you get easily fatigued. So these are just some of the symptoms and signs, but you would get the 4 marks for mentioning them. Other than that, you can talk about chest tightening. So
Just burn.
yeah, it's essentially it's chest pain, and it's associated with. It could be associated with heart problems, but in people with COPD, it's usually associated with muscle spasm and damage to the lungs. You can also get fatigue and weakness. So this is sort of related to point 4. So because they can't get the oxygen into their tissues, they feel like they have no energy, they feel weak, and they feel like they can't move, or you could think of it in other terms that they feel like they can't get oxygen to their tissues to move. So it's due to the impairment of respiration. In terms of what people with COPD actually look like, you get symptoms such as a barrel chest because their lungs are constantly inflated because their alveoli have collapsed in on each other and they can't get rid of the dead space. They also have blue fingers / face, and that's called cyanosis. Cyan just means blue. So cyanosis is when you have blue fingers and face, and that's just due to a lack of oxygenated blood getting to your extremities. So again, that's a problem with respiration. If you don't get oxygen into the body, then you're not going to get oxygen perfusing to the tips of your fingers, and that's going to make them become ischemic. If they don't have oxygen, they'll start to necrose and their color will change. So these are the main points that you can make. There are other signs and symptoms associated with COPD, but for this kind of question, you're only going to need to talk about 4. But to be safe in the exam, I would talk about as many as you know. Are you guys happy with that?
Yeah.
Yep.
All right, let's take a 5-minute break and then pick up.
Okay.
Okay. Hello?
Hello.
Hi, guys. All right, so whenever you guys are ready, just give me an explanation for question 3A.
Okay. So the normal saline solution has the same water potential as the red blood cell cytoplasm does.
Good.
Thus, water won't enter into the red blood cell or exit the red blood cell. So if the water potential were higher, then it would cause the red blood cell to explode, and if lower, it would cause the red blood cell's protoplasm to shrink.
Good. That's perfect. So you get the 2 marks for mentioning that the water potentials are equal, and the other mark for saying that there's no net movement of water. But you're exactly right. If we had a really low percentage solution of sodium chloride, then the water would move from the solution into the red blood cells, cause them to expand until they lyse. And if it was a really high percentage solution of sodium chloride, then the water would escape the red blood cells and into the solution, and it would cause them to shrink. Okay? Okay. Zero, do you want to do question B?
If the blood RBC system is just ions, I can say, for example, sodium ion.
Okay, sodium ions. And how would it cross the cell surface membrane?
Via the channel proteins.
Yep, okay, so you have a carrier protein in the membrane, or channel protein. So carrier / channel protein. And by what mechanism would it cross the membrane? So you've described the membrane component.
There is a sodium-potassium bond. Is it related to this? Because it's a sodium ion.
So the sodium potassium pump is again a membrane component, and it would come under this classification of a carrier protein. Or you could say that it's a pump protein for active transport, but again, you're talking about the membrane component. So you'll get 1 mark here for sodium ions. You'll get 1 mark here for carrier / channel protein. And to get the last mark, they'll expect you to talk about the mechanism.
Active transport or passive transport, is that?
Yeah, good, exactly. So it's either going to be facilitated.
Diffusion.
Yep. Or active transport. Perfect. That's the 3 marks. A mark.
Oh, okay. So the formula used is M equals I divided by A.
Correct.
They already give you A, which is equal to 9.5 micrometers. So
and I is 9.5 millimeters.
9.5 millimeters. Okay, so you convert 9.5 millimeters. Thank you. Into micrometers, which is equal to 95,000. Oh, did you say? So how long is it again?
9.5 millimeters.
9.5. Okay, so you'll get 9,500 micrometers.
Yeah.
And then divide that by 9.5.
Yep.
And you should get 1,000.
Good.
So that's your answer.
Yep. Perfect. So you get 1 mark for that and 1 mark for getting the formula right. Oh, sorry. Sorry. Let's scroll back up. So we're seeing red blood cells in a capillary here. And the question is to explain a feature that enables these surrounding cells to receive adequate oxygen from the capillary.
The very thin capillary, so there should be more surface area.
Yeah.
The capillary is just an epithelium cell, and it's so thin, and it allows for more diffusion.
Yeah, good, exactly. So the diameter of the capillary is small and the endothelium is 1 cell thick, and so that gives a shorter distance for diffusion of oxygen. So good, you'll get both marks for
that. Some areas in the brain known as brain-blood barriers have a type of capillary. Okay, it has no gaps between the single cells, between the endothelial cells.
Yeah, correct, good. So no gaps or fewer gaps in the endothelium. There are also tight junctions between the endothelial cells.
Okay.
And in the blood-brain barrier, the basement membrane is also impermeable. But if you make any of those points, you'll get the mark. So that's fine.
Okay. Yeah.
The structure of beta glucose in the space. How can I draw?
Yeah.
That's okay. I don't think we can easily get you guys to draw for me to be able to see it. But are you guys happy with the structure of beta glucose? Would you like me to draw out for you?
Okay, I was just say the number of carbon and how it look like. For example, it's a hexose.
Yeah.
6 carbons. With,
so wait, we're talking about beta glucose molecule here.
are you talking about the ring structure or the ring structure is a hexose.
Yep, correct.
With OH up, with the 4th carbon has the OH up in the same.
Yeah, exactly. So I won't draw the whole thing out here, but it looks like that, and the first carbon has its OH like that.
Yeah.
Are you guys happy with that?
Yeah.
Okay, good. Let's move on then.
Yes. This question. Okay. Let me. Okay. What's the question?
Let's move on. See.
The differences. Okay. Okay. So L of enzyme. Okay, L of enzyme A is higher than L of enzyme T, and then I will quote the specific temperatures and the relative activities.
So sorry, L of enzyme A is higher than L of enzyme.
Yeah, so it has a higher activity, but it has a lower temperature. So
yeah.
Yeah, and then the temperature of H for enzyme T is higher than the temperature of H for enzyme A, and then I'll quote the specific temperatures. And the relative activity of H for enzyme T is higher than that of enzyme A.
Good.
And so enzyme A is more active than enzyme T from about 10 to, I don't know, 50 something
Yeah.
Celsius.
Yeah.
And then from 50 something Celsius to 90 something Celsius.
Yeah.
It's the other way around. So T is more active than enzyme A.
Good. And comment on the optimum temperatures.
Yeah. So the optimum temperature of enzyme A is less than that of enzyme T. And I would quote the specific temperatures.
Okay, good. Yep, you'd get all the marks for that. So there's so much you can talk about here.
Yeah.
If I was answering the question, I would compare the optimum temperatures first of all.
Yeah.
Compare the ranges of activity as you did, then compare L and compare H as you did, and yeah, you get the full marks for that. So. Sure.
Both enzyme A and enzyme active. Primary structure. So this code is primary structure. So the same primary structure means that there is the same sequence of amino acids. Is that right?
Yeah, similar, not the same.
Yeah, similar. So when the enzyme disintegrates because it's a protein.
Yeah, so this enzyme is a protein. What does the primary structure dictate?
What does that mean?
No, the primary structure will influence something else for the protein. What does the primary structure influence? So you told me that the primary structure is the sequence of sequence of amino acids. Right? And what does the sequence of amino acids determine in a protein?
The type of protein.
So it will determine the secondary
And tertiary structure?
Yeah, and the quaternary structures. So, and what do the second, tertiary and quaternary structures influence in a protein?
Its shape? How?
Exactly, good. So that influences the shape, or you could say 3D structure.
Okay. It's the same as tertiary structure, right? So the 3D is the tertiary structure.
Yeah. And the quaternary is just the interaction between the different subunits. So tertiary is 3D structure of one subunit, and quaternary is the interactions between subunits. Good. So we're talking about two different enzymes, so A and T. We've been told they have similar primary structures, and you've said that that means that they probably have similar 3D structures as well, which is good. And now we're talking about enzymes. So what are the important parts of an enzyme that the structure determines?
I'm trying to see. Is it the key model, the substrate, and
Yeah, you're on the right track. So when you think about the lock and key model, you're looking at a specific part of the enzyme. Do you know what that's called?
I don't know, but I know that the substrate should fit in that.
Yeah, good. Well, if we have a substrate that looks like this.
Sorry, Ali, could you turn the volume a bit down?
Yeah, sure. Of
Please?
course.
Because I'm not able to do it from this computer somehow.
Yeah, no worries.
No, it's like, I don't know.
Oh,
Is that better?
it's because you're coming through the projector, Ali. That's no problem. It's just that it's getting picked up in the other masterclass room.
Oh, so sorry, Tom. Is this better?
Yeah, that's fine.
Okay, that's fine.
Okay, cheers. All right. So if we have a substrate that looks like this and an enzyme that looks something like this, right? And we just imagine that this is gonna go and fit in here. This part of the enzyme, do you know what it's called?
What is the first letter?
So this is the active site, A.
Yeah,
Have you heard of that before?
yeah, I have, but I forgot.
That's okay, no worries. So we're talking about, if we go back to the idea that the primary structure is going to influence the shape and 3D structure, then does it make sense that the primary structure could influence the active site? Does that make sense?
Yeah. Those have similarities, but the two exams can
Because the primary structure will go on to influence the 3D structure. And this 3D structure is what determines this shape of the enzyme. So if primary structure influences 3D structure, then it can also influence the active site. So enzymes A and T could have different shapes. So one could look like that, whilst the other looks like something like this. And these differences could be due to a change in the primary structure. Does that make sense?
I didn't get that totally.
That's okay. All right. So do you understand the concept of primary structure being the sequence of amino acids? Yeah? Okay. So if we have primary structure, in fact, I might just scroll down so we have a little more space to write. So let's say enzyme A has a primary structure, which is a sequence of amino acids that goes something like A, T, G, H, C, something like that. And enzyme T has a primary structure that goes H, A. So the only difference I've changed here is the C and the A. Now this could affect, and obviously it will have a much longer sequence of amino acids, but I've said that the only change is from this C to this A. And this is the primary structure. Now, what that might change in the secondary structure is enzyme A might have a lot of beta sheets. You know about beta sheets? So this is like when you have a secondary structure of a protein, it forms mostly either beta sheets or alpha helices. And what I've said is that enzyme A might form lots of beta sheets because of its amino acid sequence. And enzyme T, because we've made this one change, there might be a change from forming this many beta sheets to have a helix at the end, just because of the change
In one amino acid.
in
So
one amino acid. Yeah.
overall, it should have the same shape.
Yeah, it will have a very similar shape because we've only changed one amino acid.
Okay.
But there is a minor difference here, right? And in the context of the tertiary structure, when we look at that, if enzyme A's tertiary structure, I'm going to draw it as a shape because it's a sort of simpler way of representing it in a diagram. But in real life, if these were globular proteins, their tertiary structure might look something like this. We can't really think about it in that way. So instead, I'm going to draw it as a shape. You might have enzyme A looking like this, and enzyme T looking very similar, but with one difference. And if this is the active site where the enzyme binds to the substrate, and let's say our substrate looks like this, then this change is going to make enzyme T not work as well in the substrate. Does that make sense?
Yeah.
Yeah. Okay, good. So that's the knowledge that this question is assessing. So they want you to talk about how there would be similarities in the structure because they have a similar primary structure, but small differences in the primary structure can lead to differences in the secondary and tertiary structures. So you might have a very similar active site that's still complementary to the substrate, but small differences in the side chain groups might lead to different interactions between the active site and the substrate. So that could affect the thermal stability of the enzyme. So perhaps for enzyme T at 80 degrees, this active site goes on becoming something that fits better because of just changes in how the R groups interact with each other at that temperature. And that's why you get this difference in optimum temperatures. Right? Does that make sense?
Yeah.
Okay, good. The question itself is 4 marks, so it's quite a big question. In terms of how I would structure an answer, I would talk about the primary structure being similar between the enzymes and how that would lead to similar secondary and tertiary structures. Talk about how the 3D shape would then probably be very similar, but talk about how there are probably going to be some differences in the active site, and there might be some differences in the thermal stability of the enzyme because of that. And that goes on to mean that the active site will work in different ways under different temperatures. Does that make sense?
Yeah.
Okay. Let's quickly do question 5.
Okay. So production of memory cells for the first one, yes.
Yep.
Production of memory cells for the second, no.
Yeah.
So okay. Artificial active immunity and artificial passive immunity.
Good. Talk me through that.
Okay. So when a vaccine is injected into a body, it triggers an immune response which ends up with memory cells being produced, which make the secondary immune response more efficient and faster than
Good.
the primary.
Good.
Thus, you've got long-term immunity.
Yep.
So when you inject antibodies, you don't trigger any immune response, and so there are no memory cells being produced.
Good.
Additional to that, the antibodies are foreign, so they'll eventually be broken down too.
Yeah. Okay. Good. And the difference. So you've explained the production or absence of memory cells. What do you mean by artificial active and artificial passive?
Okay. So by active, I mean that we produce the antibodies ourselves, and passive, we don't. By artificial, it's an artificial process, so it doesn't occur naturally. So yeah, yeah,
Both of these individuals were injected.
they're injected. So yeah.
Yeah, so that's the artificial part, and the active part, as you say, is because we have this active process with memory cells in this vaccination, and you don't have that. It's passive because all we've been given is an antibody in this second one.
Yeah.
Okay, shall we take a break there, guys? Come back in 5 minutes.
Yeah. Yeah, we can.
All right.
Okay. Hello?
Hello? Hey guys.
Yeah, we're ready, I think.
Okay. Sarah, answer this question for me.
This outline. State the origin.
Yep.
It's from the bone marrow. Which cell exactly should I say that?
So you get the mark for saying bone marrow, which is correct. Specifically, this cell is a phagocyte, so sorry. So these come from stem cells
Okay.
in the bone marrow, and they're specifically called myelocytes. So stem cells in the bone marrow differentiate and give rise to phagocytes. So that's correct. Mark?
Yep. Um, Okay. So healthy body cells and cancerous tumor cells have different surface antigens.
Good.
So the phagocytes have a specific receptor for the cancer cells' surface antigens,
Yeah.
and by bonding with the antigen, it can phagocytosis can occur.
Good. So I've sort of drawn a little diagram here to illustrate that. So a healthy cell may have antigens here, which are distinct from the antigens here. And the idea is that the phagocytes, their receptors will bind specifically to the ones on the cancer cell. And by binding to those, they can cause the process of eliminating the cancer cells. Are you guys happy with that?
So a tumor is when cells divide and grow excessively,
Yeah.
and dead cells gonna form a tumor.
So you've said, so you get 1 point there for saying that it's uncontrolled cell division. And the second point will come from outlining either a detail of a change that occurs in the healthy cell to cause this uncontrolled cell division, or just giving me another detail about how the tumor cell, is dividing uncontrollably. So just give me one more point.
Should I mention the genetic thing or?
Yeah. Okay. So there could be a mutation. Where would the mutation be?
The mutation should be in the sequence of the gene in B cells.
Yeah. Okay. So you'll have the mutation in a gene. There are two types of genes that stop cells from being cancer cells or tumor cells. And do you know what those two kind of genes are?
No.
That's okay. Mark, do you know?
I know that an oncogene is formed when it's mutated.
Yep.
I know maybe proto-oncogene.
Good.
Okay.
And there's one other.
Tumor suppressing.
Yeah. Good.
Okay.
So in tumor suppressor gene. So in all of the cells in our bodies, we have proto-oncogenes and tumor suppressor genes. Proto-oncogenes are genes where if we mutate them, so if we somehow change them, they can become oncogenes. So proto just means able to become, and oncogene is gene that causes cancer. Whilst tumor suppressor genes are genes that are constantly stopping cells from going into uncontrolled cell divisions. So tumor suppressor genes are like cells which stop the cell cycle, or cells that cause the cell to enter a checkpoint to check that everything's okay. So if we knock out the tumor suppressor gene, then we get uncontrolled cell division. And a proto-oncogene, in terms of its function, will move the cell forward from, for instance, one phase of the cell cycle to the next phase of the cell cycle. And if we somehow mutate it, it becomes an oncogene. And oncogenes are very active in moving the cell from one stage to the other. So it will cause uncontrolled cell division. So if you're talking about a mutation causing the tumor formation, then they want you to specify that it's either going to be a mutation in a proto-oncogene or a mutation in a tumor suppressor. Okay? And, you know, that would lead to an increase in growth, increase in growth proteins, shorter interphase, rapid DNA replication. So you can add all those points down here, but the main 2 points that they're looking for is that a tumor is caused by uncontrolled cell division, and that is usually a result of a mutation in a proto-oncogene or a tumor suppressor. Is that okay?
Okay.
Okay, Mark, do you want to do this one?
So this question, I don't think it's in our syllabus right now, but so I don't know a lot about ecosystem kind of things.
That's fine. No, thanks for letting me know. If that's not in your syllabus, we'll just move on then.
Yeah. So like nitrogen fixation, everything like that, and like habitats and communities and ecosystems. It's all not part of it.
Okay, perfect. We'll just make a note of that. Okay. Right. So is this question?
Yeah, this is also not.
Also no. Okay.
No.
I'm not sure that could be the end of the paper. All right, perfect. We'll move on to the next paper then.
Yep.
Okay.
Okay. Okay.
Okay, so just have a read of this and then we'll scroll back up.
Name the stage of mitosis. Great. Okay. Prophase.
All right, 1 second. Let's fill that in. So, prophase.
Metaphase.
Okay.
Anaphase. Telophase.
Good. Right. So, start with prophase then.
Okay. A.
Yep.
Metaphase would be G. Anaphase E. And telophase B.
Good. Do you want to tell me what the other ones are?
Okay, so F and C are also anaphase.
Good.
H, I would also say is prophase.
Yeah.
Okay.
Good. And D.
Interface.
Yeah.
That's
Yeah. Good. Right. Great. Sure. Do you want to?
And then was also responsible for simply migration like a spindle at the beginning described. The spindle separate the chromosomes and bring them to the pole of the cell.
Good. So you'll get 1 mark for moving chromatids to opposite poles. And so you've explained to me the sort of final thing that the spindle does, but before it can move the chromatids to the opposite poles, what does it do?
Separate, like break the kinetochore between the chromatids. Is that
Yeah, good. That's right. So it will separate chromatids at the kinetochores. Good. So you get the 2 marks for that. The other points there, you could talk about what's happening before this happens. So at the start, the microtubules attach to the centromere or the kinetochores, and then they arrange the chromosomes at the equator, and then the spindle shortens, and that's what causes the separation and the moving. So, I mean, you get the 2 marks there, but if you wanted to talk about it from the start to the finish, then you'd start by saying, the spindle attaches and then everything it does. But that's fine. Moving on then. Mark?
Asexual reproduction and repair of damaged tissue.
Good.
Could you say so? You could also say replacement of dead tissue, or could you say T and B cells? So when they're activated.
Yeah.
Okay.
I have a question. What's the difference between repair and regeneration?
So repair is when you have, like, let's say we have a bunch of plant cells and you might have some damage to this one. Oh, that didn't really work. Let's say you have some damage here to the cell membrane, you might poke a hole in it. Mitosis, so if this cell then divided, you could then have it sort of divide and push over this one, and that one would then be destroyed, and you'd have 2 cells replacing it. And this is the sort of repair process that is constantly occurring in plant and animal cells. Regeneration of named tissues or organs is more talking about how this works on a tissue level. So this repair is on a cellular level, but regeneration is more on a tissue level. So if we think about a blood vessel that has a tunica intima and a tunica media and a tunica adventitia, if you have damage to all 3 of these layers, for instance, by a blood clot forming and damaging them progressively, then to regenerate the tissue, you need a lot of cellular replication and cellular differentiation. So repair is mostly just replication and immune cells clearing out the dead or replaced tissue, whilst regeneration is replication plus differentiation.
So where does the scar happen? Is it in repair or regeneration?
So scar tissue, it's a more complex thing that usually happens during regeneration. So because scar tissue is more a product of chronic damage. So for instance, if we have a blood clot like this, it's constantly damaging the different cell layers, you'll constantly have immune cells coming in. So for instance, phagocytes plus memory cells, which could be T or B, and then they differentiate, and you can have killer / helper cells and plasma cells. So all of these different types of cells will move in here, and they break down this cell debris, and they clear off whatever the damaging substance is. But if the substance can't be cleared, for instance, if you have an atherosclerotic plaque that creates foam cells and they don't move, they just keep causing the damage, and then the phagocytes actually add to the damage because then they die and they don't move. What you have is fibroblasts, so a different kind of cell, fibroblasts, which migrate in and they start laying down collagen and elastin and other sort of fibrous materials that are usually done in the repair and healing process. But in this condition where you constantly have damage being caused and more collagen being deposited, you get the formation of a scar. So when you think about scar tissue being formed, it's usually the process of chronic repair as opposed to acute repair. If you just have damage like a cut on your hand and you put a plaster over it, the repair process will be fast and it won't be involved with further damage, so you'll only get a very short duration where the fibroblasts come in and heal it, and so the collagen's laid down and the tissue heals properly. But if you constantly cut your hand in the same place, then the fibroblasts keep on putting down collagen and they basically regenerate the skin until you have a lot of fibrous tissue, and that's what we know as the scar. Does that make sense?
Yeah.
Okay.
Yeah, no.
None of that. Okay.
Okay. So the tissue X is ciliated epithelium.
Good.
Y is a blood cell.
Yep. Why is that?
Why is that?
Yeah, why do you say that? Why is a blood cell?
Because of the shape of the cell, and that it's quite small.
Okay, so what shape is a blood cell?
It's a it's a concave disc.
Yeah, biconcave, so it looks something like this if you look at it side on.
Yeah.
Concave is just these parts here. But yeah, you say that it's quite small. It's also in a structure. Do you know what structure it's in?
Do you mean like a vessel?
Yeah, exactly. It's in a vessel. Any idea what kind of vessel this is?
Capillary?
Yeah, what makes you say that?
Because it's quite close to like tissue fluid or other cells.
Yeah, correct. It's also seemingly one cell thick.
Yeah.
That's the cell that's making it. Okay, good. Let's move on. Right. Do you want to talk me through this?
Should I answer?
Yeah.
So first it's the cilia and the
So cilia up here.
and the goblet cells. Does that include cilia as well?
No, you're right. Goblet cells. They're different to cilia.
They
So
produce cilia.
Yep.
Myocus, I mean, they produce myocus.
good.
And what can I say first? Three ways, usually lining of the trachea, and cilia, myocus, and right. Is the myocus membrane different?
Yeah, you could talk about the sort of mucus acting as a trap. So this here I've just written down the structures that you mentioned. So the cilia can do something, the mucus can do multiple things. So you can talk about the mucus twice. So tell me what the cilia does that provides this protection against the entry of bacterial pathogens.
So the myocus, it traps the pathogens in the myocus and the cilia push them away
Good.
again to the mouth.
So mucus cells can release mucus, which traps pathogens. Cilia beats mucus away. Okay, so you've talked about the fact that the mucus can trap the pathogens, and you've talked about the fact that the cilia can then move the mucus away. Is there anything else that mucus can do that prevents the entry of bacterial pathogens?
It's the temperature. I can't see. Because no, the lining is there is a surfactant. Does that?
Yeah, okay. There is a surfactant in the within the alveoli. It's not so much in the trachea, bronchus, and bronchioles. I'm thinking about the same mucus here. So both of these points are based on mucus secreting cells, so goblet cells. They secrete mucus, and that mucus can trap pathogens. So that's the first point you've got here. And the second point that I'm getting at is that it can actually act as a barrier. So it adds an extra layer between the air, where the pathogen is, and the basement membrane that the pathogen has to pierce to enter the body. Does that make sense? So
Increase the surface area.
it acts as a physical barrier. You get the mark for saying that. So if you imagine here, if you had your pathogen, they could enter straight into these cells unless you have a thick layer of mucus, which the bacteria has to then struggle to get through. Does that make sense?
Yeah.
Good. Okay, let's do this question.
Yeah, okay. Phagocytosis is occurring.
Yep.
Explain the role of cell. Okay. So stage K is antigen representation. So the macrophage cuts up the antigen to represent its cell surface antigens on the membrane of the macrophage to activate specific to activate specific T cells, which bind to those antigens that the macrophage presents on the membrane.
Good. So your original bacteria is what we're protecting against, and you can see that it has these triangular shaped
Yeah.
antigens. What's happened is the macrophage, so that L is a macrophage, has phagocytosed,
Yeah.
and then processed. I say processed in inverted commas here because it's actually digesting the bacteria, so it's destroying it, and it's just picking up pieces of protein that are on the bacteria. And by chance, one of the pieces might also be the same part that forms this sort of triangle. So in practice, the macrophage will present all sorts of different parts of the bacterium, and by chance, one of the macrophages will present this part, that's the triangular part, and then you have this interaction between the T lymphocyte and the antigen presented on the macrophage, which causes a sort of proliferation, and then the B lymphocytes will also interact with the same antigen, and you get the production of plasma cells here.
Yeah.
And
memory cells and T killer cells.
Yeah.
Yeah.
Good.
Killer T cells.
Yeah, sorry. What time is this? This. So yeah, you get that sort of dual activation. Right. So explain how the response to second infection differs from the first. 3 points.
It will differ because the memory cell has already known the antigen, so
Good.
it produces antibodies.
Yep. So first point is that you already have memory cells, so that makes it faster.
Yeah.
Because you can raise the response quicker because you already have the cells to bind to the pathogen and cause the response to happen. To pick up the last mark, you need to talk about how this leads to faster production of either more
Antibodies.
cells, T cells, or antibody. Yeah, good. Perfect. And then you'll have a higher concentration of antibody in the blood. And so you have faster neutralization of the bacterial pathogen. Okay. Let's just do this last part and then take a break. So,
Okay. There are various ways in which. Okay. So by the number of T cells decreasing, you get a less efficient immune response, because the T cells coordinate the immune response. Yeah.
good. Second part.
Just how each of the following produces. Some
So
pathogens are covered in cell surface membrane from the host. It would be harder because it's coupled. Because there is a cell surface membrane, so it would make it. I mean,
You're on the right lines. So you've got a pathogen like this, and normally it has the antigens on it. So let's just draw the triangles. But if this pathogen then acquires a host membrane around it that doesn't have the antigens, how does that protect it?
it will not recognize.
Excellent, yeah. So no recognition. Or phagocyte that's coming along and it's trying to recognize it with its receptors just gets blocked. Good. And the last part?
if B cells don't mature properly, they won't create plasma cells with a specific antibody which binds to the specific antigen. In addition to that, the memory B cells also won't be specific.
Yeah, Good. That's it. Okay. Oh, in fact, let's take a break here, and then we'll pick up in a few minutes.
Okay. Hello?
Hi, guys.
Okay.
All right, let's start with this question.
Figure shows change that occur in grows suggest the advantage of complexity. So plasmodesmata is more about junction between plant cells. So it helps with transporting substances.
No. So this diagram here is showing two simple plasmodesmata, and these are the...
Oh, the complex one. The difference between simple and complex.
Yeah, so they're saying that as the leaf matures, you go from this sort of...
It's increasing the surface area in the complex one because
Yeah?
there are more surface area, which means easier transport.
Okay. What do you mean by easier transport?
I mean, short distance, so more diffusion is that.
Okay, so you're right in the sense that it is easier, but if you wanted to, in terms of diffusion, the describers we use is the time taken. So when you have a complex system like this, so if we look at the simple system first, the ions can only move through the two channels in one direction, if that makes sense. If you look at the really complex branched plasmodesmata, the ions and solutes could move like that, they could move like this, they could move like this, they could move like this. Essentially, you have many, many, many different pathways to move through the plasmodesmata because they're complex. So as opposed to, I mean, there is an increase in surface area here, you're correct. But in terms of diffusion, what we're doing is making it faster because there are more channels for the assimilate to move through. Does that make sense?
Okay. Yeah.
So the point is that there's faster,
Diffusion.
yeah, diffusion of assimilates. And the reason why it's faster is because there are more pathways for the assimilates to take, as I've described there. Does that make sense, guys?
Yeah.
Good. Okay, Mark?
Yeah. Okay. So okay. So when the sucrose is loaded into the phloem at the source,
Yep.
It increases the solute potential of the phloem sap, thus increasing the water potential, resulting in water flowing into the phloem at the source, increasing the hydrostatic pressure of the phloem sap at the source, which results in a water potential down the phloem by which the phloem sap travels by mass flow.
Good.
When sucrose is loaded out of the phloem at the sink, it increases the water potential in the phloem sap, but decreases the water potential outside the phloem. Thus, water flows out of the phloem and decreases the hydrostatic pressure at the sink, thus maintaining the pressure gradient / water potential gradient of the phloem.
Good. Good. That covers it all. So overall, you have a movement from source to sink down the hydrostatic gradient.
Yeah.
You guys happy with that?
Yeah.
Okay, lock and key. So we talked about this earlier. Let's go through this.
And difference between the two ways in which enzymes interact with their substrate. Induced key model or induced extend difference between the two ways in which enzymes interact with substrate.
Yeah, good. So you said
Lock model and
you mentioned lock and key and induced fit. So which one of these is lock and key and which one is induced fit?
the first one is lock and key and the second one is induced fit.
Good. Now explain the difference between the two theories to me.
So the lock and key means that substrate will only bind to the enzyme if it has complementary shape to that substrate.
Correct.
But for enzyme B, that means if the active site differ from the shape of the substrate, then the enzyme could change to adapt the difference.
Excellent. Yeah, that's correct. So with lock and key, the active site is fixed. Here we have the active site and it's fixed. And so the substrate needs to match it completely to bind, whilst in the induced fit, the active site is not fixed. So on approach of the substrate, the active site can mold itself to the shape of the substrate and then cause a fit. So that's perfect. You get full marks for that. Next question.
Okay. So Q has an alpha helical shape,
Good.
while P has a beta pleated shape.
Yeah.
So secondary structure is the structure of a protein that results from the regular folding or coiling of the protein. And
Good.
okay, and the alpha helix and the beta pleated sheets depend on the hydrogen bonds
Good.
within the protein, with the alpha helix shape being formed by strong hydrogen bonds, thus having a more coiled structure than the beta pleated sheet.
Good. And what are the hydrogen bonds formed between?
Like where exactly?
Yeah.
Between the carbonyl group and the NH group.
Good. Yeah. So you've got H bonds between these groups of the peptide bonds. And the beta pleated sheet, can you use any words to describe its nature? This is kind of just like extra marks. You will have already picked up full marks for your explanation, but there are further marks here for just talking a little bit about the nature of a beta pleated sheet.
Yeah, so it's flat, I guess.
Yep. And the sheets are stacked together in a anti-parallel fashion.
Okay.
So you kind of have you have like, if this line represented a beta sheet, it would be going that way, and then you'd have a connection, and then the second beta sheet would be going that way.
It's like DNA.
Yeah, like DNA. Okay, next question.
So carbonic anhydrase is an enzyme.
Yeah.
Catalyzes the reaction of the transformation of carbon dioxide and water
Good.
to to bicarbonate and proton or proton, right? Hydrogen.
So it's carbonic acid.
Carbonic.
So describe the role. So you've explained there what the enzyme does. So can you give me more information about the role of the enzyme then in that context?
So
where is it found?
where in the blood?
Yeah.
In the blood.
So it's in the cytoplasm of cells in the blood. Do you know which cells?
White blood cells.
Yep. Cytoplasm of red blood cells. And do you know the function of this? Why do we have carbonic anhydrase in the blood to change CO2 and water into carbonic acid?
It's because carbonic acid is acting as a buffer.
So it's involved in the Bohr effect. Do you know about the Bohr effect?
Is that regulation of the pH?
Sorry, I didn't catch that. Could you repeat that?
Regulation of the pH?
So it does cause a change in pH, but the purpose isn't to regulate the pH. It's to do with the effect of the presence of carbon dioxide changing the blood's capacity to carry oxygen.
Okay.
Mark, could you describe the Bohr effect?
Yeah, so the Bohr effect is so a high partial pressure of CO2 can results in more oxygen being released by the hemoglobin than it normally would.
And why is that?
That's due to, so you said that it catalyzes this reaction between CO2 and water into carbonic acid, which then decomposes into hydrogen carbonate ions and hydrogen ions.
Yeah.
The hydrogen ions have a high affinity to hemoglobin. Thus, they replace O2 and bind to the heme groups of the hemoglobin, forming hemoglobinic acid.
Yeah.
And yeah, they also react as, yeah, okay. That's good.
Yeah, perfect. So you got the first part right. So that is the reaction that happens, and the purpose is to sort of generate these protons. And as you said, that changes the pH. But the important part there is that it reacts with hemoglobin, and hemoglobin will normally be bound to oxygen. So you'll have oxyhemoglobin, which is hemoglobin plus 4 oxygens. And when you put it in the presence of hydrogen ions, they have a higher affinity for the hemoglobin. So that displaces the oxygen, and the hydrogen comes in and binds to the hemoglobin. And what you have is free oxygen, which can then go into the tissues. And you have the hemoglobin associated with hydrogen, which can then leave the blood and be transported back to the lungs, where it can then be reassociated with oxygen. Does that make sense?
Yeah.
Okay, perfect. I just noticed that it's 4 o'clock now, guys. So why don't we draw a line here and we can pick up with this question next week?
Yeah.
Okay. If there is anything specific that you want me to cover next week, just message me on Slack and let me know. But otherwise, any questions?
I'm good.
No question. Thank you.
Okay. Thanks, guys.
Okay.
Stop the recording now. See you next week.
See you.
Bye.
Bye.